a wonderful day today. I'm AJ Marian, along with my co-host, Dr. Devendra Agrawal, who is in charge of IACS. Now we welcome you to IACS, International Academy of Cardiovascular Science webinar series. Today, we really have a very, very special day. We are exceedingly excited about it for two reasons. First, for having First, for having one of the most eminent biomedical scientists, Dr. Toren Finkel, to speak on a very important topic that is dear to all of us as we age. And second, we have a very, very special announcement, a very unique announcement. If Natalie, could you kindly put my screen on? Say, let me see, I can share it myself. Say share. All right, this one. Allow. Yeah. Do you see my screen now?
Yes,
yeah,
sir.
we can see it.
Yeah.
All right, we have a very, very special announcement. We like to announce the most unique, the most prestigious, the most what we call among us, we call it the Nobel Prize for the Young Investigators. Truly, we are going to announce the establishment of Roberto Bolli, MD, Distinguished Young Scientist Award. This is going to be very unique, unparalleled, really, in the history of cardiovascular medicine. This is an award that will be given to the best scientists solely based on their scientific accomplishments during the annual scientific meeting of IACS. And we will send the specifics about eligibility criteria and criteria for selection of the candidates, postdoctoral fellow, residents, clinical, as research, as well as faculty member within 5 years could apply. We will choose 5 candidates for presentation at the meeting. The awardee will receive $25,000. Yes, you heard me, $25,000. It is not like any other award in cardiovascular medicine, which at most is $1,000 to $2,000, whether it is CATS Award or Young Investigator Award of American College or American Heart or IHSR. This is $25,000 to the winner of this competition. The second place finish, the runner-up will get $10,000, and the finalists will each get $5,000. So this will be basically, Dr. Bolli has been exceedingly generous and gracious to sponsor this program, and this is his initiative. Dr. Bolli is truly, from his time as the legendary former editor of Circ Research, always been dedicated and committed to advancing the career of young investigators and established several platforms at Circ Research when he was in charge to advocate and promote them. He had a program in the past for young investigator. Now he has promoted this program to a level that basically unsurpassed and unparalleled in the history of cardiovascular medicine, to give such a large sum and only based on scientific accomplishment. And this will be a 10 minutes presentation followed by 20 minutes of question and answer by the panelists, because the judgments will be on the individual. We will focus on the individual. Our sincere gratitude and appreciation and admiration for Dr. Bolli for this initiative. I will ask Dr. Agrawal, the Secretary of the IACS, whether he will like, on the behalf of IACS, make a comment, and Dr. Buddha, as the president, whether they wish to make comments, and introduce Dr. Bolli.
Thank you very much, Dr. Marian. I sincerely appreciate and echo the words of Professor Marian, who has taken the lead as the chair of this award committee. And this indeed, it is an award open to all people who are qualified from all over the world. Keep that in mind. And the meeting this year will be held in Louisville, Kentucky from 10th to 12th of September, where each of the selected ones will come and present, as Dr. Marian said that. This is indeed a legendary award, and then we would like to invite each and every one of the qualified people to apply for this. On behalf of IACS North American section, I sincerely thank Professor Bolli for his generosity and contribution in the advancement of IACS mission. Thank you very much. Dr. Don?
Thank you. I don't have much more to add. I don't have a proper adjective to describe this magnanimity, but knowing Dr. Boli for many years, I have rarely seen another person who's so committed to supporting research and young investigators. So I can see this can come only from him. So on behalf of IACS, I am once again very grateful for this initiative, and I would support this in any way possible. Thank you again.
Can you introduce Dr. Boli to say a few words, please?
So Professor Boli, first I came to know him, that was in the mid-90s, more than 30 years ago. He had a stellar career in cardiovascular research. He, as you know, has published more than 500 articles. He has been on the editorial boards of numerous journals. He changed the journal Circulation Research during his tenure as the editor-in-chief of the journal. So his contributions in the field of cardiovascular research has been tremendous, including taking stem cells to the clinic. So some of the discoveries. He has made many seminal discoveries in the field of research, and he has also been a clinician throughout his career, having managed the division of cardiology at the University of Louisville for over 30 years. So his accomplishments cannot be overstated. His generosity cannot be overemphasized. And I think we are very grateful to have him as part of IACS at this time. And we look forward to his next step of contribution, which would be this award. And we hope this would be extremely successful toward fostering the careers of bright young scientists.
Dr. Boli, would you like to make a comment?
Well, I am, thank you, Ajay and Buddha and Devendra. I am humbled and honored by your comments, and I'm very grateful to all of you for giving me this opportunity to establish an award that will identify and promote talented young scientists who will become leaders in the future. This award is different from any other young investigator award, not just because of the size of the monetary prize, but because all 5 finalists will receive a substantial cash award, and therefore they will all be winners. So we will not limit this to one person, but to 5 people, because oftentimes it's very difficult to distinguish who is really the most meritorious. And this way we will hopefully promote a larger number of talented investigators. This will be open to basic scientists, clinical scientists, and translational scientists. And so, and as Devendra said, all over the world. So hopefully we will receive a strong response. And I would like to thank in advance Dr. Marian for chairing the selection committee. I think, AJ, you will be very busy because I expect an avalanche of applications. And I would like to thank you and Devendra and Buddha for all the work that is necessary to establish this award.
Thank you, Dr. Boli. We're truly grateful to you. Really, you have been magnanimous and truly in support of young investigator. You are exceptional and exemplary. We truly are proud of you. Thank you, sir. With that, let us move to our webinar. I stopped sharing my screen, so Dr. Finkel gradually put the screen up for us. It is my distinct, really, pleasure and honor to introduce one of the world's most renowned physician scientist who truly has made seminal discoveries and contributions to our understanding on the signal and function of the oxidative stress. He was a pioneer in this area, mitochondria, consequently, cellular metabolism and autophagy in cardiovascular aging, disease and aging in general. Dr. Finkel received his undergraduate degree in physics from University of Maryland, his MD from Harvard Medical School, and PhD in biophysics from Harvard University. He trained in internal medicine at MGH, or Massachusetts General Hospital, and completed his cardiology training at Johns Hopkins Hospital in 1992. He joined the NIH as investigator and soon rose the rank of academia to position of chief of the Center for Molecular Medicine and chief of translational medicine at the NHLBI, and was in 2017 was recruited to University of Pittsburgh Medical Center at the position of G. Nicholas Beckwith III and Dorothy B. Beckwith Endowed Chair of Translational Medicine, and also to lead the Aging Institute of Pittsburgh, and was appointed as Distinguished Professor of Cardiology and Gerontology at the University of Pittsburgh. Dr. Finkel's research program from the very outset was designed to delineate the molecular biology of aging. He made a number of seminal discoveries. His classic work on reactive oxygen species as a signaling molecules put the mitochondria at the heart of aging and metabolism at the heart of aging. He followed that, focusing on the hallmarks of aging, a classic one, and classic hallmark, including autophagy, mitochondrial function, lysosomal integrity, stem cell renewal, mTOR signaling, and senescence, and has contributed in all aspects of aging to the field. Indeed, his most recent work has been on mitophagy and how to revise and modify mitophagy in order to attenuate and slow aging process. Dr. Finkel is one of the most cited scientists in the world. He has published over 400 articles in major biomedical journals, which have been cited over 11,000 times, and has earned him an H index of 112 per Google Scholar. His work on reactive oxygen species and mitochondria are among the highly cited. I'll give you one example of his article published on oxidant and oxidative stress and the biology of aging in Nature in 2000, which has been cited 13,000 times alone, a single article. Dr. Finkel has received numerous awards for his scientific contributions, including the Ellison Medical Foundation Senior Scholar in Aging Award, and he received the NIH Director's Award for Outstanding Translational Science, NHLBI Orloff Innovation Award in 2016. He was inducted to the ASCI Society, American Society of Clinical Research, and he is the Fellow of American Association for the Advancement of Science, as well as Association of American Physicians. He is a member of the National Academy of Medicine, was inducted in 2020. Dr. Finkel serves on numerous editorial boards, including the editorial board of Science. We are most delighted to host Dr. Finkel as our distinguished speaker at the IACS webinar series. He will present on senescence, lysosomes, and aging. We have assembled a distinguished group of panelists to whom we are most grateful for attending the webinar. I have Dr. Asa Burgett-Detroit, who is the leader of cardiovascular research group at the Faculty of Health Sciences at Arctic University of Norway. We have Dr. Mauricio Forte, the Division of Research of IRCCS, Institute of Neurologica Mediterranean, Cardiovascular Pathophysiology, Department of Angiocardianeurology and Translational Medicine in Italy. We have our Princess Asa Gustafsson, who was teaching earlier and was able to join us just a few minutes ago. Asa is professor at the Skaggs School of Pharmacy and Pharmaceutical Sciences at Department of Pharmacology at UCSD San Diego, and Dr. Tish Murphy, who is the chief of cardiac physiology section at cardiovascular branch at NHLBI. She might not be able to be online because government, unfortunately, is shut down, and therefore she is not capable of attending meetings unless there are health emergencies. She is not be able to join us. She might be able off and on to perhaps to sign, but we are grateful to all of you for attending today's webinar. With that, we will switch to our distinguished speaker, Dr. Toren Finkel. Now the podium is yours. We are most grateful to you to speaking at the IACS webinar. Thank you, sir.
Thank you, AJ. What a kind introduction. Can you guys all see my screen?
Yes, sir.
Okay. I'm going to talk about aging. The only one of the many disadvantages of aging, it looks like, is that I'm no longer eligible for the Roberto Bolli Young Scientist Award, which is an incredible new prize, which I'm very excited to see who will win, but it won't be me because of my age. But thank you. So I'm going to talk about aging, a little bit about a little bit of stuff that we've published over the years, and as well as some unpublished experiments. Just give you a sense of, how one might approach targeting therapeutics in the aging space, which I think will be applicable to various diseases, including cardiovascular diseases. But this will really be more aging focused than sort of cardiovascular focused. And so, I think one of the things that people always want to know is there something we can do to live longer? One thing I would suggest is to pick your parents very wisely, if possible. And so if you can pick someone like Madame Jean Calment, as shown here, who lived to 123, you'd probably be a little bit ahead of the game. How much you'd be ahead of the game is actually something that's actually quite topical right now. For many years, I think in the field, people had ascribed the genetics of longevity based on twin studies to about 10 or 20% of overall lifespan. But I don't know if some of you saw that in this week's issue of Science, a group from the Weizmann Institute in Israel revised that number, and using an updated methodology now postulate that longevity is probably about 50% genetic. So it does pay, again, to choose your parents wisely if you can. Hopefully they are long-lived. But genetics is at most 50% of how long we live. The other input is lifespan, is lifestyle, excuse me. And one of the things that we've been interested in understanding is this phenomena of caloric restriction. So it's been known for about 100 years now that if you reduce the amount of calories that an animal eats, be it something simple as yeast to something more complex like a mammal, like mice and rats, that the animal lives longer. And so, for instance, in mice, if you give an animal ad libitum diet, they'll live 2.5 years or so. If you calorically restrict them to the point of that they get just the essential calories needed to sort of for thermodynamic requirements, you can increase the lifespan, which is about 40% less than the ad libitum diet. You increase lifespan about 30 or 40% over that entire caloric restriction. So quite a robust phenomena. And the question is, how is that mediated? What are the transducers between reducing food intake and extending lifespan? And two pathways that my lab has been very interested in are shown here. One of them are a family of enzymes known as the sirtuins, which are NAD-dependent deacetylase that use NAD, which is an energy metabolite, to potentially regulate lifespan. And the other is this kinase called mTOR, which is an energy sensing kinase whose activity appears to negatively correlate with lifespan. mTOR does a lot of things in the cell. As I said, it's downstream of various nutrients and growth factors. And when nutrients and growth factors are abundant, it in turn regulates a variety of processes that allow the cell to grow and divide. That includes regulating protein synthesis, numerous changes to intracellular metabolism, and actually shutting off this process of autophagy, which we'll come back to, which is a catabolic phenomenon. And so the reason people got interested in mTOR in lifespan is a result that was done as part of the interventional testing program of the NIA, which used the small molecule rapamycin, which is an mTORC1 inhibitor, and found that when mice in particular, were given this compound, they lived about 10% longer. And so this actually became the first demonstration that you could manipulate the lifespan of a mammalian species, mice in this case, with a small molecule. And so a few years back, and we, with colleagues at the NIH, Beverly Mock in particular, characterized a mouse model where we reduced mTOR activity. And we called this model the mTOR delta-delta model. And this was a sort of unique genetic construct in which the level of mTOR expression was reduced to about 25% if you had both delta-delta alleles. So this was sort of a normal mTOR, but at roughly 25%. And the reason we went through all this sort of genetic manipulation to do this was because if you completely delete mTOR in mice, it's embryonically lethal and the animals won't survive. And if you delete just one allele, a sort of a heterozygote mouse, and reduce it to roughly 50%, there's really no effect on overall lifespan. And so it suggested that you really need to reduce mTOR protein and mTOR expression somewhere between 0 and 50% to see the lifespan extension. And in fact, this model, we think, provided that sort of sweet spot. These mice, as would be expected, are smaller than the wild-type mice as shown here, and that's consistent with the role of mTOR in regulating overall cell size and cell growth. But they are significantly long-lived. Both the males and the females that had reduced mTOR expression lived about 20% longer than the wild-type animals. And that's really, I think, a considerable increase in lifespan for manipulating one out of, let's say, roughly 30,000 genes in the mouse. Obviously, if we could do the same thing in humans, a 20% increase in lifespan would take the average individual median lifespan from, let's say, 75 years to 90 years. Again, all because you're manipulating, in this case, the expression of a single gene that acts as an energy-sensing kinase in the cell. So besides extending the lifespan, we were sort of interested in understanding sort of tissue-specific aging in this model. And that's really where the interesting, I think, analysis came about. And so we asked, in a more sort of targeted way, do these mice uniformly age slower? And I think the answer was no, not uniformly. Globally, I think in most tissues and organs that we looked at, the mTOR delta-delta mice that were going to live 20% longer at sort of any age had improved function. And so here's an example where we're looking at sort of memory and learning in these animals. And I won't go through the details of how we assess this, but when these animals are young, the wild-type mice here are shown in black, and the mTOR delta mice are shown in the open bars. When they're young, they're very good at learning and memory. And when they get old, that skill deteriorates. But you can see that it deteriorates at the same chronological age at a much more robust rate in the wild-type mice compared to the hypermorphic mTOR mice. And in fact, we looked at a number of tissues and that pattern prevailed, but there were outliers. So the bones of the hypermorphic mTOR mice actually were less had a higher age-dependent decline than the wild-type animals. And the hypermorphic mice had increased cataracts compared to the wild-type mice. So certain aging phenotypes were no better or actually worse in these long-lived species, while the majority of the phenotypes were in fact better. And so to me, this suggested that aging is not a uniform process, and that age in different tissues is probably under different regulatory mechanism, which I think is an important observation potentially, and it suggests that there may not be a single intervention or a single pill or drug to slow down aging across all tissues. And like other complex diseases, combinational therapies might be required to have the highest success at intervention. So one of the questions that arose from this study is how exactly is a reduction in mTOR activity leading to an increase in lifespan? And one of the things we focused on is that if you reduce mTOR, you actually increase this process of autophagy, which many of you know about, is this intracellular quality control mechanism in all of our cells and tissues. And the reason that we thought that that might be an important link is because work in lower organisms had linked preservation of autophagy as we age as an important determinant of overall lifespan. So for instance, as shown here in the worm, there are multiple interventions from reducing insulin signaling to reducing TOR signaling to germline removal to reducing mitochondrial function, all of which produce an increase in the lifespan of the worm. And surprisingly, when investigators have looked, all of these interventions also either maintain or augment autophagy in the worm, suggesting that they're made that autophagic flux may be an important downstream regulator of multiple pathways that regulate our overall lifespan. But obviously, these are experiments done in model organisms. Is there really evidence that autophagy declines as we get older in a species that's more sort of akin to us than the sort of post-mitotic C. elegans? And so I had a very talented postdoc while I was still at the NIH, Nua's son, who tried to develop strategies to measure, in this case, mitophagy in living organisms. And he developed this mitochema mouse. And using this, he was able to show that, in fact, that levels of autophagy do, in fact, or in this case, mitophagy, do, in fact, decline as animals get older. And so here, what Nua is doing is looking at the brain of either a young animal or an old animal. And I won't go through the details, but this orange and red fluorescence indicates high levels of mitophagy. And this is actually the hippocampus of the animal. And again, in these young, active animals, there's high levels of mitophagy occurring in this tissue. And I think you can appreciate that as these animals age, that same region of the hippocampus lacks that robust mitophagy. Again, consistent what is seen in worms, but for the first time allowing us to visualize this process in older animals. And so this all suggested that autophagy may decline as we age. And the question then arose, what is driving that impairment in autophagy? Why does mitophagy and autophagy decline with age? And we don't know the answer, but as you know, this process of autophagy requires that the autophagosome, which contains all this debris, be it dysfunctional proteins or organelles, fuse with the lysosome. And there was emerging evidence to suggest that as we age, the lysosome, like the mitochondria, really is an energetically demanding organelle. In the case of the lysosome, there's a very important pH gradient that's maintained, that's energetically demanding, that's maintained by the VATPase of the lysosome. And as you know, the lysosome itself is very acidic and has multiple hydrolytic enzymes in it. And that membrane itself is subject to constant damage. And so we became interested in understanding what are the quality control pathways that regulate lysosomal homeostasis as we age? How does the lysosome maintain its function? How does the lysosome know when it's damaged? What are the repair pathways that maintain lysosomal function? And can you begin to intervene on these pathways for therapeutic benefit? And I won't go again through the details, but my colleague Jay Tan and I, a few years back, were able to describe what we believe is a very important pathway for lysosomal mediated repair. Jay now has his own lab at our institute, and he continues to work on lysosomal biology and lysosomal function. But together, we describe this pathway, which we think is very important and again, maintaining the quality of the lysosome over time. And the first step in this pathway is the recruitment of this kinase, a lipid kinase called PI4K2 alpha. This is recruited to the damaged lysosome, and this activity of this lipid kinase converts a lipid on the surface, a phosphoinositide on the surface, PI, into a new phosphoinositide, PI4P. And so this PI4P is not normally on the surface of the lysosome, and we believe this acts in many ways to mark the lysosome as being damaged. The PI4P is also allows the recruitment of this family of proteins called ORPs. ORP proteins act as molecular tethers. On one hand, they bind to the endoplasmic reticulum. On the other, they bind to PI4P containing membranes. And when they do this, they act both as a tether and a lipid exchanger, and they exchange this PI4P that is accumulated for another lipid on the ER, phosphatidylserine. So now damage has created this sort of lipid cascade where initially we convert PI to PI4P, and then that causes the recruitment of the ER and then the exchange of PI4P for PS. And then this PS builds up on the damaged lysosomal surface, and it then activates a lipid transporter known as HTG2 that is activated by PS itself, that allows for the bulk transfer of lipids to the lysosomal membrane to allow for repair. And so this whole multi-step program we call the PIT pathway in deference to our university, but it stands for this phosphoinositide membrane tethering and lipid transport pathway for rapid lysosomal repair. And since its identification, we've now been very interested in understanding how this pathway goes awry as we age. And we've created mouse models now that we can manipulate this initial step. And we're very interested to what degree that recapitulates aging itself. And I'd be happy to come back in a few years, hopefully, and update you on those studies. But I'd like to switch gears a little bit, and because of our renewed focus on lysosome, try to think about how we can potentially manipulate lysosomes therapeutically. And I want to highlight again some of the work that my colleagues and I have been doing in this arena. Much of it focuses around the transcription factor TFAB, which is the master regulator of lysosomal biogenesis and autophagy. And TFAB is a unique, somewhat unique transcription factor that, because under normal situations, it's tethered to the lysosome. And then when lysosomes are damaged, it undergoes a variety of post-translational modifications in the cytosol that allow it to translocate to the nucleus, where it activates roughly 400 or so genes involved in this process of autophagy and lysosomal function. And I had a very talented postdoc in my group, Travis Lear, who identified recently that when TFAB goes to the nucleus, it undergoes another series of post-translational modifications that involve the phosphorylation of TFAB by a kinase called PAC2. And this phosphorylation on TFAB creates what's called a phosphodegron that allows TFAB to be recognized by this nuclear E3 ligase complex that involves a substrate adapter, DCAF7. And so what Travis's results suggested was that TFAB nuclear half-life is quite short and is regulated by this DCAF7 dependent degradation. And then the work of my that was really spearheaded by my colleagues Bill Chen and particularly Yuan Liu, were then able to sort of pharmacologically target this pathway and develop an agent which we're calling GT300, which can bind to this DCAF7 E3 ligase and block the degradation of TFAB in cells and tissues. And this prolongs it doesn't activate TFAB, but it prolongs TFAB nuclear half-life, resulting in increased overall activity. And as an example of that, you can see here that as we increase the concentration of this GT300, we can increase the number of lysosomes in cells, as shown here. So really consistent with TFEB biology, we can now use a small molecule to manipulate lysosomal number in cells. And working with my neurology colleagues, Emily Roca and Tim Greenemeyer, we saw similar things. They saw similar things in vivo. In this case, in the rat brain, they were able to dose these animals and see evidence of increased lysosomal number in this case, a region of the brain that they were particularly interested in because of its role in Parkinson's disease, both by immunohistochemistry and by enzyme activity, evidence of increased lysosomal activity. And so we've been beginning to test whether these approaches might be useful in various disease models of aging, and working again with another colleague, Dabish Sinha, who's now at Johns Hopkins. For instance, we've become very interested in seeing whether or not these compounds can be effective in diseases like macular degeneration, and early evidence suggests that, in fact, they may be, because again, many of these diseases are accompanied by impaired lysosomal function and autophagy. And so we think that this small molecule approach may be a very useful approach to a number of age-related conditions, including cardiovascular disease. Let me switch now to some unpublished results and an approach to target senescence. And again, thinking about ways that one can begin to develop new therapeutics in this area that, again, might have wide applicability across a variety of disease conditions. So senescence is an important hallmark of aging. And there's lots of evidence that, in fact, removal of senescent cells may be useful in a variety of different conditions and overall lifespans. Many of these senescent removal approaches have used genetic strategies, which really are not applicable to humans. Others have used a variety of small molecules that have been repurposed from mostly oncology applications. So this sort of dasatinib and quercetin combination, or what people call DQ, but this is actually a drug that's used in CML and has significant side effects. Navitoclax is another agent that was targeted for cancer and has a variety of on-target dose-limiting side effects that, again, make it less than ideal as a therapy for healthy elderly people as a senolytic. And so we wanted to sort of figure out ways to use genetics to identify targets that would be useful senolytic targets in sort of an unbiased fashion. Rather than starting with known agents and repurposing them, could we start with an unbiased genetic screen and identify targets that way that might be senolytic targets? So that's sort of the question that we ask. And the strategy that we've taken is to really leverage this concept of synthetic lethality that you maybe remember from your days in genetics. And so that's sort of sketched out here. So if you can think of any process that's sort of regulated by two sort of pathways, in this case, A and B, if you inhibit A, B takes over and the process is okay. If you inhibit B, A can take over. But if you inhibit A and B together, then they are synthetically lethal together, basically, because now the process is interrupted. So this pair of genetic events, A and B deletion, is a synthetically lethal pair. And this has really been leveraged in oncology, for instance, to develop drugs. So, for instance, people with BRCA1 deficiencies have an impairment in DNA repair. And if you then treat these tumors with another inhibitor of DNA repair, in this case, PARP1 inhibitors, those cells are about 1,000-fold more sensitive than wild-type cells to PARP1 DNA repair inhibition. And so that combination of BRCA1 deficiency and PARP1 inhibitors has really been leveraged in the clinic for a variety of FDA approval of PARP1 inhibitors for breast and ovarian cancer. And so this is a very useful concept, we believe. So really the question we wanted to address is, could we define a set of genes that were synthetically lethal for the senescent state? So that is, genes that when deleted were perfectly tolerated in growing cells, but when deleted in the context of a senescent cell, triggered cell death. And so to identify these genes, we engineered a cell line in which we could conditionally delete this protein TRF2, which is part of the telomere sheltering complex on the ends of chromosomes. And so this cell line had a floxed allele of TRF2 and a estrogen responsive Cre recombinase all integrated into the cell. And so when we add tamoxifen to the cell, we delete TRF2 in these cells, and that uncaps the telomeres and results in rapid growth arrest. And so I'll just show you briefly what that looks like. Again, here's a cell line that we obtained from Jonathan Alder here at Pitt. And these cells will grow normally and quite robustly. They're mouse embryonic fibroblasts. But if you add tamoxifen on day 0, what you see is these cells no longer grow, in part because you've deleted TRF2 quite uniformly in these cells, uncapped the telomeres, and basically induced this rapid senescent and uniform senescent induction in these cells in a tamoxifen-inducible fashion. Again, so you see P21 induction, you see evidence of DNA damage, and evidence of beta-galactosidase expression. And so what we've done is to leverage these cells in a genetic fashion. Basically, we start with a cell line, infect them with a whole genome mouse CRISPR library, and select for the guides, and then basically split this population of CRISPR knockout cells. In some cells, we continue to let them grow. In other cells, in a sort of duplicate set, we add tamoxifen and induce senescence. And then after roughly 12 days, we harvest the DNA from the proliferating cells, and we harvest the DNA from the senescent cells, and we compare the abundance of guide RNAs in this population and compared to this population. Again, all of these cells arose from a single population shown here. And I won't go through the details, but what we're looking for basically are guide RNAs that are particularly abundant in the proliferating cells, but are low to absent in the senescent cells. So that is, genes that were knocked out are tolerated in proliferating cells, but are not tolerated when knocked out in senescent cells. And as for some of you who've done these kinds of screens, you get lots of hits, but again, in this quadrant represent guides for genes that have that phenotype. That is, these were abundantly able to be, these were at high abundance in proliferating cells and in low or absent abundance in senescent cells. And so we went ahead and validated many of these genes. And so basically what we're asking for is, when we knock down these genes, that they kill senescent cells, but they don't kill non-senescent cells. So here's the control siRNAs. And so what we saw in many of these cases was, again, based on the screen, that in fact, when you knock down these, compared to non-senescent cells, knockdown of these genes did reduce viability, but that there was a relative reduction in viability of non-senescent from senescent, but there was effects on non-senescent cells as well. So in sort of summary, it killed both senescent and non-senescent cells when you knock down this gene, but it did more so in the senescent cells than the non-senescent cells. But that kind of phenotype wasn't ideal for a drug targeting senolytic. But there was a few examples, like shown here for this enzyme PARP16, where there was relatively little effect on the non-senescent cells, but a significant effect in terms of loss of viability in the senescent cells. And so, again, switching to human cells, we could show that we could knock down PARP16 using transient knockdown. And what this did was it, again, had really no effect on the non-senescent cells, but over the course of 4 to 6 days, induced cell death in the non-senescent cells, in the senescent cells, excuse me, suggesting that this could be a targeted senolytic. And in fact, PARP16, which is a member of the PARP family of ADP ribosyl transferases, but it's located, poorly studied and poorly drugged. But there was a group that had, in fact, developed a specific PARP16 inhibitor. They never tested it, whether it had obviously senolytic potential. But my talented postdoctoral, MD PhD student, Shahini, tested it, and in fact, it had the properties that we saw in the genetic examples I shown before, in that it had no effect on the growth of non-senescent cells, but was able to induce the death of senescent cells induced by a variety of different strategies, either doxorubicin or bleomycin. In contrast, if you take something like a PARP1 inhibitor, which is, again, well characterized, it had no effect on the ability to kill senescent cells and had sort of somewhat of a toxic effect at sort of higher doses on non-senescent cells. And so this suggested to us that, this pharmacological approach might be useful. We were able to work with our colleague at Pitt, Melanie Koningshoff, to sort of address this in human samples using precision cut lung slices. And here's an example from one donor that was had sort of very robust robust results, where we're looking at senescent cells in these human samples at baseline, and then we can treat with bleomycin in an ex vivo situation, and we see this induction of senescence in this tissue. And if we treat with this PARP16 inhibitor, we see a reduction in these senescent cells in this human ex vivo example. Other donors were not as robust as this, but we think that may be due to individual variation. But all of them saw a significant senolytic effect using this approach. I think I can just briefly summarize this last part, in that we are trying to do targeted mechanistic trials at our institute, and this is work done by Ann Newman, a colleague of mine. But we have just completed an analysis of what we are calling the RIGHT trial. Not analysis, but enrollment, what we're calling the RIGHT trial, which is targeting the IL-6 as potential therapeutic. There's a lot of evidence suggesting IL-6 is linked to both aging and frailty, as well as to cardiovascular disease. And so we were able to use an anti-IL-6 antibody that was provided by CSL Behring, and randomize patients over the age of 70 who had slow walking speed and high IL-6 levels to either this active agent or placebo. And as I mentioned, looking at a variety of both functional as well as secondary outcomes of aging, including cardiovascular function and cognitive function. And we just completed recruitment, and we're about to start analyzing the data, which we're very excited about. So let me just quickly summarize. I went through that very quickly, but I wanted to get to some discussion. Age is a single risk factor for most diseases, but I think it's very important to realize that it's regulated. I think the new genetic evidence that a lot of our lifespan is genetic suggests that that regulation is under genetic and biochemical control. And therefore, like every biological process that's regulated, it suggests that we can intervene in a targeted way. I've hopefully shown you ways that we could potentially target senescent cells in a sort of unbiased fashion, and potentially target lysosomal function, again, using small molecules. And those, and we think other approaches, we think may be a way to target a number of age-related diseases, and basically expand not necessarily lifespan, but certainly health span for our patients. So let me just stop and thank my numerous collaborators, both at the University of Pittsburgh and other places. I particularly want to highlight Shahini, who was a very talented MD PhD student who did that synthetic lethal screen. Bill Chen and Ewan Liu, who pioneered the TFEB small molecule, and Ann Newman, who did the right study that I mentioned. So I'll stop there. Again, I know that was quick, but I wanted to get the panel and questions.
Thank you, Toren. We are truly grateful to you for your comprehensive presentation covering in-depth molecular biology of lysosomes to clinical trial of IL-6. We're grateful to you for your fantastic presentation. With that, the presentation is open to question by the panelists as well as attendees.
Yeah. Can I ask a question?
Dr. Bowley,
Yeah.
legendary editor.
Yeah, thank you, Jay. Thank you, Toren. Very elegant, exciting work. So I'm going to ask you a very practical, simplistic question. As you said, we are all coping with aging, and we all try to find ways to slow that down. What would you think about many quote anti-aging supplements end quote, such as resveratrol or NMN that modulate allegedly sirtuins or other pathways? Is there any solid evidence that they prolong longevity in humans?
I don't think so. I think I think for the most part they're probably not harmful. Although you see people on 50 or 60. People who take supplements tend to take lots of supplements, and then you see people on this sort of polypharmacy where they take 50 or 60 of these supplements, and then you see really problems. I personally take none of those. I don't think any of the evidence is strong enough to warrant any of those interventions. The only thing I personally do, and again, I don't think the evidence is very strong, is I do sort of time-restricted feeding. So I actually try not to eat breakfast or lunch and sort of eat all of my food sort of in the evening. But again, the evidence in mice is pretty good for that. But I think there's a lot of reasons to think I think if you base immunological work off of mice, so many things work in the mice that have no bearings to humans. So I think at this point, the only thing you can say is, pick your parents wisely, probably don't try to eat reasonably and moderately and exercise, or probably the strongest thing you can say. Everything else, I think, is questionable.
Okay, thank you.
Interesting. So we have two questions, one by Dr. Priyantash Gerha, my colleague here. Dr. Gerha would like to know what would be a potential issue for chronic lysosomal activation. Also, how does lysosomal and autophagy activity overall correlate with species that live long as compared to short-living animals?
Great questions. So there, like everything in biology, there's probably a window in which you need to operate. So in general, for instance, TFEB, there are certain tumors where TFEB undergoes molecular rearrangement and is expressed at 30 or 40-fold higher than under normal conditions, and that appears to drive tumorigenesis. So too much TFEB is probably a bad thing. Same thing with lysosomal acidification. Although in many tissues it declines, in hematopoietic stem cells with age it actually increases, and that increase is part of the decline. Is bad for those cells. So I think the answer is that I think for all the therapies in aging, what you're trying to do is not to overshoot, is to sort of correct back to sort of the youthful more level, but obviously that's a very tall task to do pharmacologically, and so that's part of the challenge. I don't know how to compare. The second part of the question is quite interesting. I don't know how to compare lysosomal function across species. I don't know of a good assay potentially to do that, but that's an interesting question. How to measure autophagic rates or lysosomal function as a function of lifespan species, basically. I don't know how to do that, and I don't know if it's been done, but it's a good answer. Good question.
Great. So before we go on with the attendees, including Sydney and Dr. Cheng and Coco's and Dr. Junichi Abbey questions, so many questions coming up. Let me give a chance to our panelists. Asa from Norway has raised her hand. Asa, please activate your microphone and kindly ask comments.
Thank you, Toren, for a really nice talk. I was wondering, since it's all about lysosome fitness that gets reduced during aging or senescence, is there anything known about the level of autophagic lysosomal reformation versus TFAB-induced lysosomal biogenesis in senescence or aging?
Yeah, so for many years, the classic hallmark of senescence was lysosomal beta-gal expressions. That's the sort of histological stain, and people sort of didn't think about that, I think, in a sort of mechanistic way, why that is true. And so very recently, it's been shown that senescence is accompanied by TFAB nuclear translocation, and that TFAB is required for lysosomal biogenesis in the case of senescence. And then if you block TFAB from going to the nucleus, what happens is that cells that are stressed now undergo apoptosis rather than going to senescence. And so that it appears as if, it's a very interesting question, why cells, when they're damaged, they have a choice between dying or senescing, and what determines that balance. And it looks like if you can expand your lysosomal compartment and basically deal with the damage, presumably through autophagic recycling, then you'll senesce. And if you don't have that response, you tend to die. And actually, a graduate student in my lab has now sort of characterized a little bit more detail how that TFAB activation occurs. And so she's been able to show that it occurs through this process not of macroautophagy, but of non-canonical autophagy, a process called chasm. And so that when cells are senesced, actually the chasm is activated on the lysosome itself, and that's the mechanism by which TFAB is activated. And if you block chasm, you get the same phenomena where cells now die in response to stress as opposed to undergoing senescence. So yeah, that's sort of what we understand at this point.
Great. Now we have DK, Dr. Agarwal.
Thank you, Dr. Marian.
IACS.
Yeah, so Professor Finkel is brilliant information, brilliant talk. Thank you very much for sharing with us. I have two very quick questions. Is there any relationship between the human size and longevity? And second thing, is there any difference in regards to the aging by calorie restriction in the evening, in the afternoon versus during the morning and daytime? As you mentioned, and also Dr. Boli is on the same boat that he does not eat breakfast or lunch, he only eats dinner. So is there any difference if somebody restricts in the evening versus during the daytime?
Yeah, I don't know the answer to that question. It's not an area, I mean, for me, it's more practical. It's like, when I'm finished work, I want to eat so I don't know if there's benefit, to eating your calories when you're awake versus, in both cases when you're awake, but, in the morning versus evening. It's a great question. I don't know to what degree that's been looked at in animal models. The problem is, mice are different in terms of day, night, and light, dark than us. So it gets confusing. I don't think there's any human data to one way or the other. I forgot your second. So I can't answer that question. I forgot the first part of your question. I'm sorry.
Human size and longevity. Is there any data?
I've asked my colleague Ann Newman this, and I've always thought, there's definitely, there's definitely species within a species like dogs, there's definitely a correlation where smaller animals, smaller dogs live longer than bigger dogs. And then across species, bigger species like elephants live longer than smaller species like mice. So there is some correlation. I think in humans, I've asked Ed Newman, and it's not very robust, that there is no evidence that larger people have shorter lifespans, which is very comforting to me because I'm 6'4", and I was afraid that I would suffer. But she said that that's not true. That it hasn't.
All right, getting back to attendees, Dr. Swetny Chen would like to know if you have to prioritize, do you think lysosomal capacity is more limited by biogenesis or by functional quality of the lysosome?
Yeah, that's a great question. I think I think capacity is probably, I think as we age, it's not a huge effect on the number of lysosomes, but their quality. It's same thing, for instance, with mitochondria. It's not a reduction in number, but quality. So I think it's really the ability to maintain that pH gradient with time, and the acidic environment in the lysosome, which is again a energy dependent process, I think is probably the thing that varies most as we age, not necessarily our biogenesis capacity.
All right, fantastic. So we have a question from, forgive me for misspelling your name, Zhao Kang Chen. It says, very exciting talk, Dr. Finkel. Can you comment on the cell death pathway responsible for the effect of PARP16 knockdown in senescent cells?
Yeah, that's a great question. We're not entirely sure. PARP16 is on the endoplasmic reticulum, and it's involved in maintaining proteostasis, and it's in the few papers that have been published on it. And what we observe is that if you knock down PARP16, there's really no effect of proteostasis in normal cells. But again, the combination of senescence plus PARP16 results in this dramatic increase in protein aggregates in the cell. And so we think that's what's driving cell death, that there's some specific requirement for the proteostasis effects of PARP16 in the senescent context. And again, it's that combination of senescence and the absence of PARP16 activity that leads to this massive protein aggregation in the cell, in the senescent cell, and we think subsequent cell death.
Very good. We have another question by Sai Koka. Would like to know, does PARP16 inhibitor DB008 affect other PARPs, considering PARP family has highly conservative domains?
Yeah, that's a great question. It was developed. We didn't develop it. It was developed by a group out in Oregon. In our hands, it does have selectivity for PARP16. It's about 5-fold more selective against PARP16 than PARP1. It's a covalent compound that leverages a unique cysteine residue in the PARP16 binding domain that's not present in the other PARP family members. But again, some selectivity, but not 100-fold.
Very good. Thank you. The next question for our neighbor at MD Anderson across the street, Junichi Abe. Great talk. How do the extratelomeric functions of TRF2 contribute to genome stability?
Yeah. So again, we just used that model as a genetic model for senescence, and really didn't, we didn't really characterize what exactly TRF2 is doing when it's what the overall effects of TRF2 deletion in that model. We do know that it results in telomere uncapping and telomeric fusion and rapid growth arrest and cell senescence. It may have other effects, but we didn't characterize them in detail.
Very good. Thank you, sir. The next question from our distinguished faculty here, Laila Ruhi. In post-mitotic tissues such as the heart and brain, do you think mTOR inhibition slows aging through fundamentally different mechanisms compared with proliferative tissues?
Yeah, that's a great question. You know, I think people who have used rapamycin, for instance, and looked at tissue-specific effects, did see the most profound, I think, effects in sort of cognitive function in terms of its age-dependent effects. And so you may be right that there may be more pronounced anti-aging effects in non-dividing tissues, but I do think that we saw effects in other tissues as well. It'd be hard to ascribe the relative efficacy, but it's certainly possible that mTOR inhibition is fundamentally different in non-dividing cells than it is in dividing cells.
Excellent. Thank you. Next question from Brian Robinson. Given the microbiome can impact each of the processes you mentioned, what is the evidence that microbial input can augment aging phenotypes? Is there a role for probiotics and microbial manipulation in therapies?
Yeah, this is not an area that I'm that well versed on or know that much about. So everything, I don't have much of great insight to say. I should say that the only thing that I could say that's of any relevance is that that last study we talked about, which is the IL-6 inhibition trial, that there is this generalized inflammation or inflammaging in elderly people, and the source of those inflammatory cytokines is obscure. Some people say it's derived from the what's called the SASP, the senescent associatory secretory phenotype. So as you accumulate senescent cells in your body, they are producing the IL-6, and that's why levels go up. But other people say that it's really coming from the gut and the microbiome, and that a leaky gut is really the driver of inflammaging. And so in that context, I think the microbiome may be very important as a driver of age-dependent phenotypes. But again, it's not an area that I particularly know that much about or have worked on.
Thank you, sir. We have a question from Frederica Morelli-Berg. Great talk. Thank you. Does segregation of mTOR to the lysosomal membrane have anything to do with lysosome fitness and autophagy?
Yeah, absolutely. So, I mean, again, one of its downstream effectors is TFEB itself. So, under normal conditions, mTOR will phosphorylate TFEB, and that phosphorylation event retains TFEB in the cytosol. And then, when the lysosomes are stressed or there's nutrient deficit, mTOR activity is inhibited. That results in the dephosphorylation of TFEB, which then allows it to go into the nucleus and activate autophagic and lysosomal pathways. And so mTOR is a critical regulator of autophagy and TFEB biogenesis, basically.
Thank you, sir. And final question. This is from Dr. Sweti Chen. I have another question. In aging cells, what do you think fails first? Lysosomal degradation capacity itself, or the signaling system that decide what gets sent to the lysosome?
Yeah, I think I think it's a hard question to answer. I think if you look at old cells, the classic thing that's been noted since the 1800s is sort of this lipofuscin pigment that is sort of aging pigment that represents undigested material, proteins, lipids, and other things in the lysosome. That's what lipofuscin is. And so to me, that suggests that things are getting there. They're just not being degraded once they get there. So, I mean, that's an 18th century answer or 19th century answer. We probably could address that more robustly, but my sense is, the problem probably lies in the function of the lysosome, not in their numbers or targeting of substrates per se to the lysosome.
Thank you all very much. Thank you, Dr. Finkel, for a fascinating talk. You can see that there's a lot of enthusiasm for your presentation. We truly are grateful to you for your fantastic presentation. And thank you, the panelists, Dr. Assad and Dr. Mauricio, and the other Assad had to run to a meeting, and of course, Tish couldn't participate. We're grateful to you all for participating. Thank you, Dr. Boli, for establishing the Dr. Boli, MD, Distinguished Young Scientist Award. We're grateful to you all. This was a fascinating webinar, indeed, mesmerizing day for all of us. And from the bottom of our hearts, thank you all very much for your contribution to this academic activity. Our sincere gratitude to you all. Thank you.
Thank you. Bye-bye.
Bye-bye. Bye-bye.
Thank you. Bye.
Bye-bye.
Thank you, sir.
Bye. Thank you.
Bye-bye. Thank you, DK.
Yes, sir.
Thank you, Professor Natalie.
So Natalie will
Bye. Thank you.
AJ, Natalie will send you the webinar before we release it. So
Excellent.
you look at it and approve it. Thank you.
We do it. Hey, by the way, did my last slide show during the question and answer
Yes.
series?